Hello students, welcome back to another chapter of this agronomy series, the principles of agronomy series. So today we are going to discuss about mineral nutrition, manures and fertilizer. So first we'll see what are the mineral nutrients plant required, how they are being taken up by the plant and a lot of things. Okay. So, for survival of the plant, there are so many factors that is important. I mean, different biotic factors are also affecting the growth and development and yield. Different abiotic factors also affect growth, development, and yield. So, one of the abiotic factor that is nutrient that the plant is mostly getting from soil, except one or two, like carbon dioxide, I mean carbon and hydrogen, oxygen. Except these two, three, uh, most of the nutrients they are taken up, and all the mineral nutrients they are taken up from the soil. So how these mineral nutrients and whether all the nutrients, all the minerals present in the earth, they are essential for the plant, which nutrients are essential, how this essentiality is being established for a plant, what are the form of uptake of the mineral nutrients, all these things we are going to cover in this particular lecture. So far, we have covered so many things about agronomy. You can watch them from our previous lectures. We have so many lectures uploaded on our channel. Now, coming to mineral nutrition, this is nothing but the process of mineral nutrition is nothing but the process of absorption, translocation, and assimilation of nutrient. That means when we are talking about a plant, suppose these are the root system, this is the canopy. Okay, so basically this is the soil uh, where from the below layer, I mean there is the root is always underground the layer, so except some plant root like crop root, breast root and all. So basically what is mineral nutrition? Mineral nutrition is the absorption of the nutrient through the root system, the translocation of the nutrient through the root system, and assimilation, that means in taking part in different biomolecule process and biomolecule conversion it into different biomolecule or enzymatic activity. So the whole process is known as mineral nutrition, and we are going to discuss about all these three. Now, how many nutrients are essential for the plant, and what is that essentiality? I mean, whether all the nutrients available or the all the minerals or all the elements that is available in the periodic table they are essential for the plant? Absolutely no. Okay. There are so many, I mean, plant require more than 60 elements for its survival, but not for survival, for its growth. But among those more than 60 nutrients, only 17 are called essential nutrients. Only 17 are called essential nutrients. Why they are called essential nutrients? Because they fulfill the essentiality criteria that was proposed by Arnon and Stout in 1939. Okay, and redefined by Arnon in 1954. So essentiality criteria that was proposed by Arnon and Stout in 1939, and it was redefined or re-established by Arnon in 1954. So what are these essentiality criteria? There are three essentiality criteria. If any nutrient fulfilling these three essentiality criteria, then that nutrient is called essential nutrient for plant. Okay, so based on this essentiality criteria, 17 nutrients are regarded as essential nutrients. So what are these essentiality criteria? The first criteria is plant cannot complete their vegetative or reproductive stage without the without that particular nutrient or deficient of that particular nutrient. So plant cannot complete the vegetative or reproductive stage of life cycle due to deficiency of that particular nutrient. Okay, this is the first criteria. If the nutrient is deficient, then plant cannot complete its vegetative or reproductive life cycle. Second criteria is deficiency can be corrected or prevented by supplying only that particular nutrient that is deficient. Okay, suppose one nutrient is deficient, we cannot fulfill that nutrient deficiency through other nutrient. Okay, so that was the second criteria. The third criteria is the element has direct influence on the metabolism of the plant. So that element we are talking about, that must have direct involvement in the metabolism in the plant of the plant. So these three criteria, which are called essentiality criteria, proposed by Arnon and Stout in 1939 and redefined by Arnon in 1954, if any particular nutrient is fulfilling these three criteria, then that is called essential nutrient. Though among these three essentiality criteria, only criteria 1 is universal. Okay. Criteria 2 and 3 has some exceptions. Okay. So what are these three 17 essential nutrients? They are again grouped into basic nutrients, okay, then primary macronutrient, secondary macronutrient, and micronutrient. So what are the basic nutrient and why these are called basic nutrient or structural nutrient? They are carbon, hydrogen, and oxygen. These three are called basic nutrient because they constituent around 95 to 96% of the dry matter of the plant. That means major nutrient. These are the major main structural element, nutrient. Carbon around in every plant you can get carbon around 40 to 45%, oxygen around 40 to 45%, hydrogen around 4 to 5%. So around 96% uh, basically constituent by this carbon, hydrogen, and oxygen. Then coming to primary macronutrient. Primary macronutrient are N, P, K, nitrogen, phosphorus, and potassium. They are called primary macronutrient because their requirement is more than 1000 ppm. Their requirement is more than 1000 ppm. This primary macronutrient, their requirement is more than 1000 ppm. That means more than 1000 milligram per kg of plant dry matter. Then calcium, magnesium, and sulfur. These are called secondary macronutrient. Okay. Now micronutrient. These are iron, manganese, copper, zinc, boron, molybdenum, chlorine, nickel. Okay, these are called micronutrients. So this micro and macronutrient classification, it was proposed by Loomis and Sol. Loomis and Sol. Okay, they proposed macro and micronutrient concept. So more than 1000 ppm macronutrient, less than 1000 ppm micronutrient. Now another form of nutrient is required by some specific plant, that is called beneficial nutrient. Okay, beneficial nutrient means functional nutrient. Okay, so functional nutrient consist of essential nutrient and plus beneficial nutrient, right? Essential nutrient, we just talked ki 17 essential nutrients are there, but what is beneficial nutrient? Their action is actually not specific. They do not fulfill these three criteria of essentiality, but they have certain beneficial role in certain specific plant. Like suppose rice. Okay, rice is also called a siliceous plant. It requires silica for its stem stiffness. Okay, and from preventing lodging and preventing disease pest infestation in the stem, it requires silica. So that is a beneficial nutrient for our rice plant. Then mostly halophytes, they require sodium for for their metabolism. For their survival in the saline condition. So that is a beneficial nutrient. So beneficial nutrients are four, that is sodium, cobalt, vanadium, and silicon. Including 17 essential nutrients, these 21 are called functional nutrient. Functional nutrient, remember 21, which consist of 17 essential nutrient and sodium, cobalt, vanadium, silicon. Okay, these are beneficial nutrient. Now, functional nutrient in plant metabolism, whether its action is specific or not. Okay, so basically this is not definition of functional nutrient, this is the definition of beneficial nutrient, right? Next one is nutrient classification. One classification we just covered, like macronutrient and micronutrient classification. Another nutrient classification is there based on physiological and biochemical function. So based on physiological and biochemical function, how nutrients are being grouped, and according to this criteria, physiological and biochemical functions, Menzel and Kirkby, they classified nutrients into four different groups. Four groups are basic structural nutrient, then energy storage, transfer and bonding, then charge balance, then enzyme activation and electron transport. So basically, which nutrients are under the basic structural nutrient we have already covered, like carbon, hydrogen, oxygen, which cover 95 to 96% of the plant biomass. Then some nutrients, they are helpful in energy storage, transfer and bonding, like nitrogen, sulfur and phosphorus. Some helps in charge balance in the plant, like potassium, calcium and magnesium. Some helps in enzyme activation and electron transport, like mostly micronutrient like iron, manganese, zinc, copper, boron, molybdenum. Okay, so this is how Mengel and Kirkby grouped all the nutrients, essential nutrients, into, based on physiological and biochemical function, into four groups. Now, based on the requirement of these particular nutrients, like primary nutrient, secondary nutrient, or macronutrient, micronutrient, this is how we can develop a graph. I mean, suppose this is the yield. Okay, this is quantity of nutrient required. So when we are increasing the macronutrient concentration, then the yield increases, or macronutrient availability, the yield increases and remain maintain plateau. But when a micronutrient, we are talking about micronutrient, their yield also increase, but after plateau their toxicity is also more prominent. Okay, so for macronutrient it is more or less showing asymptotic curve, and for micronutrient it is more or less showing parabolic curve. Now, uh, so when a plant is taking up nutrient, uh, based on uh, from which area it is taking up the nutrient, that area is basically, from nutrient point of view, it is called forage area, right? From nutrient point of view, it is called nutrition point of view, the root area from where plant, I mean surrounding area, surrounding to root, from the area where plant takes up the nutrient, that is called forage area. And this forage area is much larger or much wider in case of mobile nutrient than the immobile nutrient. So for mobile nutrient, you can see here, suppose this is the root system, and for mobile nutrient, total area surrounding to the root, that is called forage area, because the nutrient is mobile in the soil, like nitrogen, like boron, like sulfate, like manganese. Okay, or I mean chlorine. Okay, so these are highly mobile nutrient in the soil, so there the forage area is larger. Okay, because from any part of the surrounding soil, the nutrient can translocate to the, I mean diffuse to or translocate to the root system, and it can easily absorbed by the root. But for immobile nutrient, it is closely adhered to the root and root hair and the root system. So the forage area is very limited for immobile nutrient. Okay, are you getting my point? This is the forage area for mobile nutrient, this is the forage area for immobile nutrient, because the as the nutrient is immobile in soil, only that particular nutrient that is closely adhered to the root system, that can be absorbed by the plant. So that is in case of immobile nutrient. So whenever we are applying any fertilizer, we can broadcast or we can randomly throw the fertilizer in, if the fertilizer is nitrogenous fertilizer or potassium fertilizer, which is mobile in the soil. But in case of immobile nutrient like phosphorus, we have to place it near the root zone so that its absorption will be maximum. So that point we have highlighted here, like for highly mobile nutrient, the entire root zone is forage area. So again, if we'll classify the nutrient based on its mobility in the soil, then mobility in the soil, highly mobile nutrient, less mobile nutrient, immobile nutrient. Here you have to see all the nutrients, like which are having negative charge or anionic nutrients, or which nutrient are taken up by the plant through anionic means, they are highly mobile, because the soil system is electrically negative. Okay, so when the nutrient is negatively charged, it repels the nutrient, so it is not particularly adsorbed or absorbed in the soil clay complex, so it is highly mobile. Exceptional is manganese. Okay, then less mobile nutrient, all the cationic charge nutrient, all the nutrients, they are taken up in the form of cation, they are less mobile nutrient because they can be attracted to the soil clay complex, and the mobility is hindered. Then immobile nutrient, only two words are like phosphorus and zinc. They are immobile in soil. Now next one is nutrient mobility in plant. Based on the nutrient mobility in plant, nutrients are classified into highly mobile nutrient, moderately mobile nutrient, less mobile nutrient, and immobile nutrient. Highly mobile nutrients are N, P, K, magnesium, and molybdenum. Moderately mobile nutrient is zinc. Less mobile nutrient is iron, manganese, copper, sulfur, and chlorine. Immobile nutrient, boron, and calcium. So this is the nutrient classification based on mobility in soil and plant. You have to remember, and from this we will see how the deficiency symptoms, their symptom differ in plant from lower leaf to higher upper leaf or bud region. So that is based on this mobility in the plant. Then based on the chemical nature of the nutrient, some nutrients are taken up as, some are cationic nutrients, some are anionic nutrient. Like cationic nutrient like potassium, calcium, magnesium, iron, manganese, zinc, copper. Anionic form of nutrient like nitrogen, phosphorus, sulfur, chlorine, though nitrogen is taken up in both cation and anionic form. Then metallic nutrient like potassium, calcium, magnesium, iron, manganese, copper, zinc, and non-metallic nutrient nitrogen, phosphorus, sulfur, boron, molybdenum, chlorine. Now, what are the factors affecting nutrient availability in the soil? What are the factors influencing the nutrient availability in the soil? So there are so many factors that affect directly or indirectly the nutrient availability in the soil. One is parent material. Based on the parent material, which nutrient will be available, which nutrient will be more, which is less. That depends on the parent material, because parent material is the ultimate source of the nutrients from where nutrient is released. Then pH, mostly the micronutrient and phosphorus, they are influenced by soil pH. Okay, so for ultimate or optimum availability of the nutrient, pH must be optimum. Then microorganism, they convert the nutrient from mobile to immobile form, and immobile to mobile form. So basically, their availability or microbial activity is also affecting the nutrient dynamics in the soil. Soil organic matter, because soil organic matter is the storehouse of different nutrients, and if the soil organic matter is proper, then microbial activity will be proper, then nutrient availability will be optimum. Then addition of fertilizer, which fertilizer we are adding, based on that plant will get different type of nutrient. Soil temperature, as different enzymatic activity and soil activity, soil reaction, it depends on the soil temperature. So soil temperature is also having significant influence. Then moisture, mostly nutrients that are taken up in the ionic form, and when ionic form of nutrient available, when the moisture is proper. So proper moisture condition is there in the soil, because nutrient when it gets solubilized in the soil water, then only it can form cationic or anionic form. Then aeration, okay, most of the nutrient they depends I mean air or oxygen for its mineralization, or that means from unavailable form to available form, right? So these are then cropping system, what type of cropping system or crops the farmer is taking, based on that also nutrient availability will be differ. So mostly if the farmer is taking monocropping year round, then nitrogen deficiency would more likely to occur. Then rhizosphere colonizes. See, when we are talking about rhizosphere, that is nothing but the plant root system. So root system colonizes around 23 fungal species but dominated by penicillium species. Now transport of nutrient to the root surface. Okay, so there are two types of theory based on which the nutrient transformation happen to the root system. One is soil suction theory, another one is contact exchange theory. So soil suction theory is followed by mass flow and diffusion. Contact exchange theory is followed by root interception. So basically these are three methods through which nutrient is being absorbed or taken up by the, I mean, nutrients reach to the plant root and nutrient exchange happen. So mass flow means when there is transpiration gradient, suppose this is the plant system again, these are the canopy. So what is root, I mean transpiration pool? Transpiration pool means suppose there is, let's say, soil, what is the whatever the water potential is in the plant, the water potential is more negative. I mean more water is demanded by the plant. More water is again from the stem, more water is demanded by the leaf. From the leaf, more water is demanded by the air or atmosphere. So when the atmosphere is unsaturated, it demands water from the canopy of the plant. So it pulls the water from the canopy through transpiration process. Then when there is water deficiency in the canopy, as this water is removed to the atmosphere, then it pulls water from the xylem vessel from the stem. Then stem pulls water for its need from root system. Root pulls water from the soil. So this is a passive system. Okay, it is governed by the diffusion pressure gradient. Okay. So through this gradient, while water entering to the root system and moving to the canopy, some nutrients they are soluble in the water, they also enter through the root system. So this process is known as mass flow process. Then diffusion, it is basically a process through which from the higher concentration of the nutrient moves to the lower concentration, that is diffusion. And mostly phosphorus and potassium, this type of nutrient, they are taken up or they are coming to the root system through diffusion process. Then mass flow, root interception, they are basically helpful in case of immobile nutrient where the root itself moves towards the nutrient, and when there is contact of nutrient with the root happens, then there is exchange happen. Okay, exchange of nutrient from the soil to the root system, it happens. That is called root interception. Now soil nutrient in the soil, that is a dynamic system. Okay, so how this nutrient dynamics happen, like initially the soil nutrient is in unavailable form in organic form, then it coming to intermediate form, then labile form, then soil solution form. This soil solution, when the nutrient reaches here in the soil solution, this is actually available for the plant uptake. And this soil solution nutrient, this form is known as intensity factor. So how intensely nutrient is available, the labile form, and before that whatever it is available, that is called quantity factor. What is the quantity of nutrient available? Always intensity factor is less than quantity factor, and intensity factor is helpful in case of plant nutrient uptake. Okay, now absorption of nutrient, it is depends on several factor, again internal factor and external factor. What are the internal factor that affects the absorption of the nutrient? One is cell wall. What is the density of the cell wall, what is the lignin content of the cell wall, how permeable the cell wall is, all these factor. Like then types of cell and its development, then transpiration, rhizosphere is also affecting the nutrient demand or nutrient absorption. Then external factor like oxidation reduction state of the nutrient, then concentration of the nutrient in the soil, then moisture content of the soil, pH, aeration, temperature. All these factors they affect the absorption of the nutrient. Then how this nutrient absorption happen? There are two mechanisms. One is active absorption, another one is passive absorption. In active absorption, there is energy is consumed for absorbing the nutrient. So when there is proper mass flow is not happening, proper, I mean, nutrient is not coming through the water stream, then the plant has to involve some kind of metabolic energy to absorb the nutrient, that is called active absorption. Passive absorption is delivered through, or it is carried out through diffusion pressure gradient, as I have already mentioned, and it is basically the transpiration pool. So that is passive absorption without spending any energy. Now we just discussed about nutrient absorption, and then the nutrient is translocated in the plant system. Okay, once the nutrient is absorbed through the root, then it is translocated, that means it moves inside the plant. So how it is translocated? Initial entered through root system, then it moves to cortex, then xylem vessel, then leaves, then there is assimilation of the nutrient. Assimilation means when the nutrient is taken up by the root system as and translocated in the plant in ionic form, it has to be uh, used in the plant system. I mean, it can act as a carrier or cofactor in an enzyme, or it may be take part in any biomolecule synthesis. So that has to be used in the plant system. So that is called assimilation of the nutrient. So basically assimilation is nothing but the metabolic transformation of inorganic nutrient to organic form. Okay. So basically, literal meaning is to make it similar. As of the biomolecule present in the plant system, it has to make it similar. So that is basically the assimilation part. Then assimilation of nitrogen. First one is, let's discuss about assimilation of nitrogen. So how this assimilation of nitrogen happen? So nitrogen, as you know, it is mainly taken up by the plant in case of nitrate form or ammoniacal form. So when it is taken up by the plant through nitrate form, it combines with NADH and H plus in the cytoplasm, and there the enzyme activates the nitrate reductase, which reduce this NO3 form to NO2 minus. NO2 minus. So that is basically nitrate reductase. Nitrate is reduced to NO2 minus. Then again NAD plus and H2O. Then nitrite reductase act here, and this process, the first process is happening in cytoplasm. Basically, the nitrate reduction is happening in cytoplasm. Then nitrite reduction is happening in chloroplast. So nitrate reduction through nitrite reductase, and it convert it into NH3. NH3 ammonia or ammonium. Then plus along with water, it converts ammonia plus glutamic acid plus ATP, and this glutamic acid with the help of glutamic synthase, it forms glutamine plus ADP, and this is in mostly in the root cell. Then glutamine in presence of alpha ketoglutarate acid or glutamic acid synthesis happen. So it forms 2 glutamic acid, which is taking part in as a base material for synthesis of other amino acid, and it is called as transamination process. So through transamination process, from glutamic acid it forms different amino acid in the plant. And you know, amino acid are the building block of protein, and protein is helpful for the structural stability in the plant. Okay, so this is how nitrogen is assimilated in the plant. First, nitrate is converted to nitrite, nitrite converts to ammonia, ammonia converts to glutamic acid converts to glutamine, then glutamine to 2 glutamic acid, which is used by used as in the transamination process to form different amino acid. Now, nitrate assimilation takes more efficiently in case of C4 plant than C3 plant. C4 plants, the carbon dioxide reduction in bundle sheath cell and light reaction and nitrate reduction in mesophyll cell. So in different cell as the activity is happening, so it is more efficient than C3 plant. Now coming to assimilation of phosphorus. Assimilation of phosphorus. So here the phosphorus, inorganic phosphate that is taken up by the plant, that associated with ADP, adenosine diphosphate. Then respiratory energy is involved, where oxidative phosphorylation happens to form ATP. Okay, this is in root. Then ADP plus inorganic phosphate, radiant energy, and in chloroplast, this is in root, and in chloroplast ADP plus PI, radiant energy or photosynthetic pigments phosphorylis that converts to ATP, and this happens in chloroplast. Okay, so respiratory energy is used in root and radiant energy is used in chloroplast or leaves. That is the basic difference. Otherwise, inorganic phosphate is attached to ADP and it converts to ATP, and that ATP is used in different metabolic activity. Now assimilation of sulfate. Again, sulfate is, sulfate is taken up by the plant in SO4 2 minus. So sulfate is converted to sulfide, sulfide to sulfide, then cysteine. Cysteine is a sulfur containing amino acid. Other sulfur containing amino acids are methionine and cysteine. Okay, methionine and cysteine. These are sulfur containing amino acid, and this process is happen in chloroplast, and this amino acid helps in synthesis of different proteins in the plant. Assimilation of other cations, like mostly in coordinated bond, it forms chelate form, and they absorbed and translocated in the plant system, and they assimilated in as in different enzyme cofactor as well as plant biomolecule. Now coming to so for this lecture this much. Next we'll cover the function of nutrient and deficiency symptom of different nutrients, how they looks, so that you can rectify the nutrient deficiency by applying different nutrients and by identifying the deficiency symptom. So if you have any doubt regarding this lecture, then let me know in the comment section so that we can prepare another lecture with detailed clarification or detailed analysis. So keep reading uh, and keep signing till the next lecture. Bye bye.